In my part of the presentation, I will briefly talk about what the high-level topics are that we're going to cover and discuss how you might benefit from participating in the program, what might be the possible outcomes. I'll introduce you to some of the mentors that will be helping you along the way and show you some examples of students that have completed this program and what they have done with the information that they received. So as you all know, we live in the age of data. And specifically in biomedical research or in biology, there's a growing source of data. And one data in particular, called next generation sequencing data, is becoming kind of the foundation of a lot of the different research topics that are being done in biomedical research and agriculture and biotechnology, et cetera. So my background is actually not in bioinformatics, but I came into bioinformatics by working with Pine Biotech and the different collaborators that include multiple research universities, biomedical research projects and consortia. And I've had the privilege of training and working alongside experts and students in many different institutions here in the US. So as a result of that, I've seen how it's really unpredictable to know who becomes an expert and who leaves and who kind of chooses bioinformatics as their topic. And for a lot of people, what I've discovered is it's not clear what bioinformatics actually is and how is it applied in a variety of domains. So my first part of today's session is to explain why bioinformatics and what is bioinformatics and how data in biomedical research is actually used to advance the application of insights from such analyses to advance precision medicine, biotechnology, agriculture, etc. So first of all, kind of a historical background. Since the 1950s, when the DNA was first discovered, the structure of DNA was clarified, we've seen how such information has led to new discoveries, starting from the sequencing, annotation, and discovery of the human genome and the genes that are responsible for different proteins, to technologies that make such data easy to produce and eventually to use in large-scale initiatives like Genomics England, the Broad Institute, different medical centers that actually use insights from genomic data to categorize, diagnose, and treat patients. Now, the basis of that is that when we understand the code and we understand what the code actually produces, so how nucleotide sequences form amino acid sequences that are the basis of proteins, we can start looking at variation of that DNA code that is responsible for some specific phenotype. Or in this case, in this example, you can see sickle cell anemia, actually a change in a particular amino acid leads to a change in a cell structure, right? And so that kind of an ability to detect changes has led to a number of very clear applications. So for example, drug metabolism, how different individuals metabolize different drugs, whether they will respond or not respond to treatment, identification of mutations in cancer cells, identifying genetic predisposition or disorders like sickle cell anemia, inherited mutations, rare genetic diseases, as well as drug resistance and different pathogens. And these examples are all in the context of biomedical research and medical application in the context of precision or personalized medicine. Because what happens in the medical context is today clinicians have to deal with such information to actually make a better description or characterization of disease for a patient. And one of the things that we now are starting to see is that you have these panels of specific genes, specific proteins, and you can now identify mutations that are responsible for very dangerous diseases like cancer. Now, as researchers continue to study these, they actually understand that some specific genes in the genome are responsible for the most serious effects. So, for example, tumor suppressor genes, oncogenes, DNA repair genes, they accumulate mutations that are most dangerous. One example is the TP53 mutation that is a gene, P53 protein, tumor protein P53 is encoded in that gene. And what that mutation does is it can actually alter the structure of that protein and make it not functional. And if it's not functional, it's a tumor suppressor gene. So then it does not suppress the tumor growth because it does not respond to the damage in the DNA and leads to accumulation of mutations. So finding such mutations can be critical for accurate and precise and effective cancer treatment. In cystic fibrosis, you have mutations that are responsible for the functioning of the CFTR channel, which is responsible for getting the right kind of chloride ions out of the cells. And if it's not functioning, it leads to accumulation of mucus and can be very dangerous. So knowing how to find these mutations and understand their function is clearly important. And not only is this important for individual mutations, this is also important for large-scale rearrangements, like copy number variation, repeats that expand a particular region. And one way to find those variations is to perform genome-wide analyses that take thousands of samples for clear phenotypes and start understanding what are the statistically significant patterns that occur in a large proportion of the population. Such studies have led to discoveries, including tuberculosis drug resistance, which is a disease that is affecting millions of individuals around the world. If someone can monitor the participants and make sure that no one, thank you, if you could keep them unmuted, thank you. So drug resistance, not only to individual drugs, but multiple drugs is starting to become a huge problem. And we see the same in the world of viral pathogens. So for example, the Ebola outbreak and how that leads to tracking of transmission and vaccine design, but also the current pandemic of COVID-19. We hear about variants of concern all the time. So all of these project examples, all of these examples in precision medicine, in population health, genetic predisposition to disease, all of these have been transformed by the availability of these different types of sequencing techniques. So in the program, what we will try to do is introduce you to how such data is generated. What is the process of acquiring actual digital data from a biological sample? And then in that data set, how do we work with different algorithms, computational algorithms, to understand mutations that are pathogenic, neutral, that are maybe not very bad or bad? And how do we determine the effect of these mutations in the genome? So how do we actually categorize them? How do we understand their impact on the phenotype? And what are some examples where you can find the clear association between individual mutations and the phenotype? And so that is really what bioinformatics is all about. So bioinformatics is about taking information or data, taking methods of computational analysis, statistical methods, machine learning, analytical methods, and applying them to biological data sets, applying them to data sets where we have biological samples, conditions, clinical conditions, et cetera. And so the program is going to tie together these three different domains in a way that you can quickly see the application of bioinformatics to a number of different domains. So the OmicsLogic programs are going to help you not only understand how these things work, but also practice and apply them. And so in the context of this program, these resources that were already mentioned by Sri Gauri are going to be critical to getting you from maybe not knowing a lot about bioinformatics to getting an understanding of what bioinformatics is and how it applies, to eventually starting to use it, maybe in your own project or a group. And so here, we're going to try to cover a variety of different types of examples or projects that are case studies that you can use to really understand and develop a sense of you personally discovering something on your own using some of these tools. Now, I imagine that most of you here have understanding of what bioinformatics is, maybe not a lot of experience, and that is really who the program is for. Now, I want to specifically highlight two projects or two resources on the OmicsLogic portal where you can get started. So even if you're here for the first time, now that you have registered, you can go to the OmicsLogic portal and find two specific courses. One is called Introduction to Bioinformatics, and another one is called Bytes and Molecules. So Sparsh or Srigori, somebody maybe can, someone can maybe post the links. So if you have an account and you've signed up, again, the sign up is free. Those are two introductory courses that will get you started. They will get you into this context. They will explain at a high level the different applications, introduce you to our community, the way that things are organized, and you will really get a good sense of why bioinformatics is it for me and what can I do with it. Okay, thank you, Tash. So Introduction to Bioinformatics is that first course, and the second course that I wanted to mention is called Bytes and Molecules. So again, if you search here, Bytes and Molecules, it is course 2. And this course is different from the Introduction to Bioinformatics because it is a little bit more detailed and actually introduces a lot of the concepts that are going to be useful for what we do in this program. And that includes terminology, or what are these terms like SNPs, variants, gene expression, profiles, etc. It will give you a sense of the relationship between code and structure and how things work in the cell. And then it will introduce you to specific types of data that could be acquired from next generation sequencing, so genomics, transcriptomics, and metagenomics, and talk a little bit about maybe things that you haven't thought of how bioinformatics is actually used to study precision medicine or astronaut health in space. Now, also a big component of bioinformatics, as I mentioned, is the application of data analysis techniques and tools to large data sets. And coding is a critical component. So as you've seen, one of the critical elements is to understand how and why, and then have the practical skills. And so the second option that was mentioned in the beginning, so the first one was genomics in the virtual lab. The second one was coding or biomedical data analysis in R and Python. So in this course, you will already get an introduction to coding in R, which is a popular method of analyzing different types of biological data, where you will have access to different examples of just getting to know what coding is, why do we need coding, and how to read, produce, to load the data, things like that. So that's going to be very useful because one of the practical examples here is the ability to actually run code and practice fixing the code that is given to you without having to take too much time of yours to actually learn everything from scratch. So we're not going to learn things like hello world. We're going to jump into how to load data. And this data is going to be biological data, or the type of data that you are expected to use in the domain of bioinformatics. And once we get into this, you will see that you will start to appreciate syntax. You will know how to load and produce simple plots that are informative. You to interpret these plots. And then as it becomes more and more complex, so here you will find assignments, those will introduce you to more and more complex methods of analysis, including analysis of complex data, analysis using complex methods like machine learning and deep learning, and also ability to integrate multiple sources of information to actually answer real research questions. So along the way, as you go through this coursework, you will realize that a lot of this coursework is sourced from experts in the field. And experts in the field are people that have a long history of maybe doing research or using such skills in the domain of biotechnology and pharma. And getting to know these people, you will realize that they all have a rich history and a path of how they got to where they are today. So to briefly introduce some of these people, I want to highlight a couple examples. And those examples are going to demonstrate kind of the quality of the people that you will be exposed to.